Okay, this is the BIOL 213 Vertebrate Zoology Skeleton Lab, and we will be following the theme, which is broadly around the axial and appendicular skeleton. Within the axial, we'll start with the skull and the story that occurs here, and then we'll deal with teeth before moving to the axial vertebrae, and then down into the appendicular skeleton and the skeleton overall. So we start here with the cranial skeleton. The first component is the chondrocranium, which is shown here for dogfish, which is a cartilage chondrocranium, and that becomes a neurocranium in the ossified form in latter vertebrates. Okay, so that's a brain case, and really the primary function is about supporting sensory systems. It ultimately gets overlaid by a dermatocranium and also supported by the splonchocranium. So let's meet the splonchocranium next. Originally, this is the pharyngeal basket and pharyngeal slits of a cephalochordate. And then in the vertebrates, we see a derived form in the agnathans in the brachial basket of a lamprey. So that's homologous to those pharyngeal basket. And in these guys, they have a lingual cartilage, which is really the first arch of the pharyngeal basket, and the rest of it's forming their brachial basket. These guys also have some cartilages which are the nascent form of a chondrocranium, so that's the lamprey. We then move forward and look at what happens in the shark, which is representative of an early gnathostome. Now these guys, they have modified the first and the second arch into the mandibles and the hyoid. And this is important because you can see the mandibles here, and those are suspended by the hyoid. And so these got a type of jaw articulation called hyostyle, which we'll see in a minute. Now, you then move forward into the early tetrapods, and we see a big change. What starts to happen is that mandible gets replaced by a dentary. And that's important because the mandible is also called a Meckel's cartilage, and it changes. One end of it becomes a little bit which we call the articular, and the top part of the mandible, that one becomes a quadrate, and these are overlain over the top with dermal bone in the specifically in the lower mandible. It's called dentary bone. So that's all good, but what happened to the hyoid suspension? Because that's now, we don't really have a suspended jaw. It's instead, there's just like the skull, and then there's a lower jaw articulated to that. So the hyoid has become a separate bone called a columella, and we'll pick that up later when we look at ear ossicles. That's what's going on with these guys. What's happening to all the other cartilages, which were the parts of the pharyngeal basket? So, we've looked at what happened to 1 and 2 arch. The rest of them, well, ultimately, those are supporting the throat, the hyoid, and then in particular supporting the larynx. And we see that there in the mammals, which are a bit more derived. Okay, I'm going to jump to a separate section to look at what happens next. So we're going to jump over here to the ear ossicles, because I've just talked about how we've moved from a hyostyle to a fused jaw. And I've talked about how this dentary bone takes over from the earlier mandibular bone. That's all good because the hyoid arch becomes a columella. In mammals, we see a change again. Now, the remnant of the mandible, which is called Meckel's cartilage, completely gets replaced by the dentary. And so what happens is the little bit of Meckel's cartilage that made the articular joint is going to become a malleus, and the little bit that made the top that became the quadrate is going to become the incus. And the columella, which was originally that hyoid, hyomandibular, it's now called a stapes. And so that's that story. And you'll notice that we now have this different type of articulation. It's a complicated story, but it's basically how you go from gill arches, with numbers 1 and 2 being jaws, to a different jaw articulation for a better processing food on land. And then in the mammals, different jaw articulation again, where we've really got rid of those 2 gill arches, and they've become 1 and 2 become the ear ossicles, and all the rest become components of the throat and the larynx. And instead, we now have dermal bone forming the jaw. That's the story going on there. And it's all about an improved articulation for food processing, and the ear ossicles are all about shifting the connection between the jaw and the ear so that the ear is completely separate. Because otherwise, when you chew, you hear crunching noises and you can't hear properly. Okay, now we can go to the dermatocranium. Recalling we've done chondrocranium, splongenocranium, dermatocranium. And the theme here is that we had dermal armor in the early ostracoderms, which essentially gets reduced. And it becomes the outside skull with lots of little bones. But as you move forward, this is a bowfin. It's an early fish. If you look at mammals, they've really got fewer bones and they're fused into a more composite skull in this guy. And now you can see here that in this guy, there's a dentary bone overlaying the mandible, Meckel's cartilage. And in these guys, that Meckel cartilage is completely gone and you just have the dentary bone. It's the only form of jaw. So that's the dermatocranium of these guys. Okay, we're going to roll forward a little bit more, and we're now going to look at fenestrations. It's the next story in the dermatocranium. So 3 types of fenestrations, which are holes in the skull to enable muscles to pass through, and apsids don't have one. So if you look at these guys, the jaw muscles have to attach to the inside of the skull. We have diapsids, which is the reptiles, and here the jaw muscles pass through and can connect to the outside of the skull through these 2 sets of fenestrations. And then in mammals, you can see this most clearly. So in these guys, they have only 1 fenestration, synapsids, and it comes through here, those muscles of the jaw, and attaches to the outside of the head. So you have the temporalis muscle, and then you have the masseter muscle down here, and those are very large. And they can be very large because they are not confined to being on the inside of the skull. And you can kind of see that in this. Attached to the inside of the skull, transitioning to attach to the outside of the skull with a hole for that muscle to pass through. Some other useful things to note. These guys, unlike humans, have got this arch here, sagittal crest, which is for the attachment of the muscle, so they can have really powerful jaws, and that's that story of increasing chewing power. We'll see that again coming up in a couple of other stations soon. This bit's called the zygomatic arch, and this is called the postorbital arch. If you look at some carnivores, they're missing the postorbital arch. They still have the zygomatic. Don't worry about the lack of that postorbital. It's to do with the mechanics of the orientation of the eye, and it's favorable in some. We carry on with our dermatocranium, and we're now going to go and have a little look at the secondary palate. I'm going to start with a bullfrog. You can flip these guys over, and they have choanae, which are holes that go from the nasal passage to the mouth, right at the front of the mouth. That means if they are eating food, they will simultaneously be struggling to breathe. So the 2 are mutually exclusive. However, if you move forward to a mammal, you can see that the choanae move right to the back of the mouth because they've grown a secondary palate bone. So let's see. Here's a lizard, which is a halfway house, and you can see the choanae halfway back. And then if you look at the mammal, dog, you can see the choanae right at the back of the mouth. Don't get confused by these. Now, those ones there are actually the nasopalatine duct. Those are for the vomeronasal organ, so they're a little bit different. So look here. Here's the choanae, and there's those other holes at the front. Don't confuse the 2. So that means that this is the secondary palate. There'll be a soft palate extending further, and this allows you to both chew and breathe at the same time. Again, we're seeing that same theme repeated. Okay, I covered the ear ossicles earlier, but just in case you're wondering, there are those resources. We have the columella picked out on the early amphibian skull as an example. And then here's our malleus, incus, and stapes just over here. So I believe that we've got there, if I'm correct, we're going to have the malleus is the one in the middle, the incus on the right, and then the stapes down the bottom. Recalling that they are the, I believe, the quadrate, articular, and then the columella, or hyoid. So these 2 from the first arch, this one from the second arch. Okay. We go over here. Now we've got teeth and dentition in the mouth. There's 3 forms. You can either have your teeth on top of the jaw, which is acrodont. You could have them stuck to the side of the jaw, which is pleurodont, or you could have them in a socket, which is thecodont. So early fish and sharks, they're actually acrodont. Later fish, bowfin and others, and reptiles, they tend to be pleurodont, and then a thecodont would be something like this elk here, or an alligator perhaps. You might want to look at that when you look at an alligator skull. Okay, so we also describe the teeth in terms of whether they are replaced once or not at all. So this one's not at all. It's a beaver. They just grow teeth throughout their life. These are ongoing growth. We have humans, which are a good example of one which replaces their teeth once. And then here's a polyphyodont, which is replaced throughout the life. We're diphyodont. Beaver's monophyodont. We can also have a nice little look here at the dentition in terms of whether it's the same or different. So if you have a look at an alligator gar, teeth are all the same. A lizard, teeth are all broadly the same. This one might actually be a mud puppy. Yes, it is. So when you move to a mammal, we are heterodont. So the others are homodont, mammals are heterodont. Different teeth for different functions. Here's a good example of that. You've got your grinding teeth and your incisor teeth. And you'll recall that you need to know how to do a dentary formula. So just remember that you'd be saying the top divided by the bottom. So this guy here is going to be I33 for incisors, C11. Then I think for premolars, I think we're going to have P44, and then for the molars, M22. That's me guessing on a dog. I'd need to double check that, but I'll be looking for you to be able to do this. Placoid scale is from an earlier lab, but remember it's homologous to early bone. And then we get to our axial vertebra. So we'll start off by looking at the notochord, which remember is not a vertebra, but this gets ossified. In lamprey, there are some remnants or components of the arches. We're not seeing them there in this specimen, but they do have them, which is why they are a vertebrate. And then we see actually vertebrae proper, bone vertebrae. Here's some fish vertebrae, and then we've got some bird vertebrae, and we have some mammalian ones around as well. Here we are. So, oh, no, those ones are bird as well. Mammals over there. There you are, there's a cow. And the story here is that we have different types of articulation depending upon the type of movement the animal does. And I particularly want to emphasize the bird vertebrae because these ones are heterocoelous, there's that term, which enables them to bend both left and right and up and down. So it gives them much more mobility. And you can see here, there's an example of an atlas and an axis, which is a special innovation with the atlas atlas evolving in amphibians, so they have some mobility on land, and the axis evolving later in reptiles. Okay, there they are again. Whoops, they're back to front. That's an atlas, that's an axis. Make sure you get that right around. Human skeleton, useful for seeing the specialization of the different regions of the vertebral column. That's our theme now as we move through terrestrial animals. They have to have much more support in certain regions of the body. For example, the lumbar, the sacral region, and then cervical needs to be still fairly mobile. So you can see these differences. And I'd like you to know the different regions. Like these are spines for often shape or muscle attachment. You have processes for muscle attachment. You have a centrum, processes, centrum, spine, and you have this hole down here, which is for your dorsal nerve cord. Okay, these guys don't really have very clear. The point of that hole is it's part of the arch, the neural arch. Okay, we go forward. We see a fish skeleton, homologous structure to not homologous structure. Very, very much the same as we move down the whole of the vertebra on the spinal column there. You can see the early ribs, and these guys have only got one process. These guys have got two for the ribs. So that's a transition there depending upon whether they need to support the internal organs. It's about the main point there. We then introduce the pectoral and pelvic girdles. So you have the pectoral and the pelvic, which are not articulating with the skeleton in a fish. Actually, these down here in a teleos to the pelvic, they've really, really moved forward on the skeleton. But when we move to the terrestrial vertebrates, we do see the articulation with the skeleton which is best illustrated on our cat. So here we have the sacral and the ilium actually articulating together. Back up at the front, our pectoral girdle is not fully connected. It floats free but is supported by the scapula and the clavicle. Okay, comparison here, cat, which is a running animal, a walking, running, jumping. This guy is sartorial, so cursorial, sartorial. And if you're looking at the frog, you can talk about the fused urostyle, number 3 there, and then other components such as the powerful jumping legs. When you look at the cat, you can see elongate limbs for running, you can see a tail for balance, and you can see nicely the differentiation of the vertebrae. Cervical, we've got sacral, lumbar, thoracic, and, caudal, sacral, lumbar, thoracic, and cervical, and how those are specialized for different functions, from elongate to support long tail for balance, sort of sturdy to support the body weight and for attachment of the ilium, and then we have this more flexible cervical vertebrae. Finally, we look at some changes to our articular skeleton. Snake, they've lost it, become very, very much homologous, or I keep using that, homo for the same throughout the skeleton and the vertebrae. We move to a bird and a bat, both of which evolve flights. We see modifications to the appendicular skeleton, which is subtly different depending upon how they spread the membrane in a bat using finger bones, or in a bird support the feathers. And then we have a bird skeleton, which is conveniently showing many structures that are adaptations to flight. So obviously we have the flexible cervical vertebrae, but others, we have the sternum with a keel that supports the flight muscles, together with the synsacrum and the fused thoracic vertebrae, which support a sturdy structure for muscle attachment and flight. We have a pygostyle, which is a reduced tail region and fused to support the tail for steering. So there's some good examples of their bones for supporting flight. Okay, that is the end of this lab. And if you did ever want to reference it, this is that story of what happens with the splonchna cranium and its interaction with the dermatocranium. So going through from an agnathan, you'll see Meckel's cartilage is the first gill arch, hyoid is going to be the second gill arch, and you can see that change through an early gnathostome fish with its hyostyly jaw articulation, to an early tetrapod on land, which has become autostyly, and we now start to see a dermal skeleton involved in the jaw. And then finally we see the loss of the splonchna cranium, first and second arch, from the jaw, and instead those are fully incorporated into